Hello everyone, my name is Dr. Minda and I'm going to complete the series also the eyeball. So it's able to analyze the form of light, the intensity of light, and the color that's reflected from objects. So it contains a fibrous globe, which is tough to maintain the shape. It has a lens that focuses on the image, and it has photosensitive cells that are actually able to collect information and process, and then the nerves that will transmit this visual information to the brain. So this is actually the structure of the eyeball. So you have an anterior chamber here, the pupil, the iris, the lens suspended by the zonules here onto the ciliary body. Then you can appreciate that the eyeball has three layers: an outer sclera posteriorly, and the outer part, then followed by the choroid, and the retina is the innermost. And then optic nerve exits, and the nerve contains central retinal artery and vein within it. So there are various parts you need to pause and so again, this is the anterior aspect. There's the cornea there, and that's the aqueous humor. Okay, and then this is the lens with the suspensory ligaments, and then the three layers. So you have your sclera followed by choroid and the retina. That's the lamina to the central retinal artery and vein. Vitreous humor occupies the space posterior to the lens, and anterior to the lens you have the aqueous humor. So it has three layers. So the fibrous tunic has the posterior sclera and anterior cornea, and the cornea is transparent, so forms part of the optic apparatus. After the fibrous tunic, you have the vascular tunic, or the uveal tract. It has three parts: the choroid, ciliary body, and the iris. So the choroid is for nutrition. The ciliary, when the ciliary muscles contract, they're able to affect the ligaments that are supporting the lens. So that's how they help with the optic apparatus diaphragm to regulate the amount of light the eye. So basically that's so the eye. So you have photoreceptors, and the retina covers the ciliary. Okay, so it really functions more of the nutritive function. Then we have the fibrous tunic now. The sclera, it's opaque. It's the outermost portion on the posterior part. It's opaque. It's avascular, no blood vessels. It has dense connective tissue, collagen, and elastic fibers with melanocytes. Okay, and the deep part. Then it gives the eyeball its shape, and it provides site for attachment of extraocular muscles. So anteriorly, what happens? The sclera merges with the cornea at what we call the sclerospar, and that's the point where ciliary muscles insert. Then posterior part of the sclera, we have the lamina cribrosa where optic nerve perforates. Then we have the cornea. The cornea is colorless. Remember, we need light to pass through. It's transparent and it has 5 layers. So these 5 layers, we usually call them A, B, C, D, E, which we are going to discuss. Remember, the cornea, we need it to be transparent, so it has no blood vessels and no melanocytes. You don't need pigmentation, you don't need blood vessels, but it has rich sensory nerve supply, because remember, the corneal reflex needs to protect the eye from anything that wants to come and fall into the eye. So what are the 5 layers? A. Anterior epithelium, which is stratified squamous. B. The Bowman's membrane, made up of collagen fibers and no cells. C. Is the corneal stroma, made up of connective tissue, and you have regularly oriented collagen fibrils. Regular, because you need it to be transparent and allow light to pass through. D is the decimate membrane. It's homogeneous, so it has collagen filaments in 3 dimensions. And then E is the endothelium, which is the posterior epithelium. The anterior epithelium was stratified squamous, posteriorly is posterior endothelium. So that's the A, B, C, D, E of the cornea. So that's the epithelium, Bowman's membrane, before you go to corneal stroma, the decimate membrane, and corneal endothelium. Again, so that's the cornea. So basically epithelium, then you get to the Bowman's membrane, which is the anterior limiting lamina, before you get to the stroma, then posterior limiting lamina, which is the decimate membrane, before you get to the posterior endothelium. So that's the structure of the cornea. So what the cornea has endothelium posteriorly and epithelium on the surface, and this helps to maintain the transparency of the cornea. And how does this work? Because you have sodium being transported towards the apical surfaces, and sodium, chloride, and water, they follow together, and that way you're able to maintain the corneal stroma in a dehydrated state, and that keeps it transparent. So the dehydrated state, along with the regular orientation of collagen fibrils, keep the cornea transparent, and the fact that it has no blood vessels and it has no melanocytes. Then we have the corneoscleral junction, which is also called the limbus. Corneoscleral junction is the transition from the transparent cornea to the opaque sclera. Remember, the two of them form the outer portion of the eyeball. Posteriorly is the sclera, which is opaque. Anteriorly is the transparent cornea. So the transition is the corneoscleral junction of the limbus. It's highly vascularized, but the cornea is avascular. So how does the cornea get its nourishment? Diffusion from the vessels at the corneoscleral junction, and also the fluid within the anterior chamber will have nutrients that will leave it via diffusion into the cornea. At the limbus, this corneoscleral junction, we have trabecular meshwork, which we call the canal of Schlemm. This canal of Schlemm helps to reabsorb fluid from the anterior chamber. So it drains the fluid from the anterior chamber. So the canal of Schlemm actually communicates externally with the venous system. So this is what we are discussing. Between cornea and sclera at the junction is the corneoscleral junction, or the limbus, and that's where you have the trabecular meshwork, where you have canal of Schlemm, and that's where aqueous humor will be reabsorbed back into the venous system. So you can see this is the canal of Schlemm. You can appreciate the trabecular network, and that's where the reabsorption of aqueous humor from anterior chamber into the venous blood will occur at the corneoscleral junction. So after the corneosclera, we move to which is the fibrous tunic, we move to the vascular tunic, or the uveal tract. We said it contains the choroid, then the ciliary body, and the iris. Choroid, ciliary body here, and the iris. So those are the three parts of the vascular layer. So the choroid is highly vascularized. It has loose connective tissue. So you have fibroblasts, macrophages, lymphocytes, immune cells. Then you have collagen and elastic fibers. Choroid is pigmented, so you have melanocytes, so it has a dark color. Even the iris is pigmented. So the blood vessels in the inner part of the choroid form the choriocapillary layer, and these are the ones that supply the retina with nutrients. Then we have also a thin hyaline membrane that separates now this choriocapillary layer from the retina, and that's what we call the Brooks membrane. So the choroid has a choriocapillary layer with blood vessels, and that's separated from the retina by the Brooks membrane, which is a hyaline membrane. Then we go to the ciliary body. Ciliary body is just an inward extension of the choroid at the level of the lens. Then we have projections from the ciliary body, which are short extensions towards the lens, which are called ciliary processes. So ciliary body basically is just made up of loose connective tissue and smooth muscle cells. The ciliary processes are extensions of the ciliary body, and they contain dense capillary network. Then we have ciliary epithelium. This is epithelium of the ciliary body. It usually produces aqueous humor, and aqueous humor is just plasma filtrate from the capillaries. After it has produced the plasma filtrate, this plasma filtrate enters the posterior chamber. Now we have tight junction between cells of the ciliary epithelium, and these form the blood aqueous humor barrier. Blood aqueous humor barrier is formed by tight junction between the ciliary epithelial cells. So ciliary bodies have ciliary epithelium, and these cells have tight junction between them, and that forms blood aqueous humor barrier. Then we have zonal fibers. What are zonal fibers? You have fibers from the ciliary processes that extend to the lens. So they're the ones suspending the lens. They're called suspensory ligaments of the lens. And we have ciliary muscles. Ciliary muscles actually attach to the sclera, and they are able to stretch the ciliary body. So when the ciliary muscle contracts, what happens? You tend to pull on the tension of the zonal fibers, and when they relax, you reduce the tension, and that thickens the lens. And when you thicken the lens, you're able to focus on objects that are close by or near, and that's what you call accommodation. So when ciliary muscles contract, you stretch the ciliary body, and you stretch the zonal fibers and stretch the lens for far vision. But when the ciliary muscles when you have less tension of the ciliary muscles, the lens now thickens and you're able to see things that are near, and that's what we call accommodation. So then we go to the iris. The iris is now extension of the choroid and it partially covers the lens anteriorly. The anterior surface of the iris is irregular and rough. So the iris has three parts. You have the discontinuous layer made up of pigment cells and fibroblasts. Then it has a poorly vascularized connective tissue layer, and this has few fibers and many fibroblasts and melanocytes. Then lastly, there's a vascularized layer that contains loose connective tissue. So the posterior surface of the iris is smooth and it has two layers of epithelium. The inner epithelium actually has melanin, so it's heavily pigmented, and the pigmentation prevents passage of light into the eye except through the pupil. So when the iris is pigmented, light will only pass through the pupil. So all the pigmented portions will prevent light from passing through. So the amount of pigment actually varies in people. So some people have greenish blue, gray, or brown irises. Albinos have no pigment in their iris. So it tends to look pink because of reflection from the blood vessels in the iris. The iris is vascularized. So the iris contains concentric smooth muscle cells. Okay, and these are what form the sphincter pupillae muscle. So they constrict the pupil. Then, so the iris has dilator pupillae and sphincter pupillae. Dilator pupillae muscles are innervated by sympathetic nerves, so they will cause dilatation of the pupil. Sphincter pupillae muscles are innervated by parasympathetic nerve. They will cause constriction of the pupil. So this is the lens here, and this is the iris, and that's the ciliary body. The ciliary body is lined by, it has extensions, so you have your ciliary processes. Okay. So the lens is biconvex. It is very elastic, and the elasticity is actually lost with age because as you age, the lens hardens. So we have three histological parts of the lens. There's a lens capsule, an outer lens capsule. Below it, we have subcapsular epithelium. And after the subcapsular epithelium, we have lens fibers. The lens does not have blood vessels or nerves. So the lens capsule is thick, homogeneous, and refractile, and it contains high carbohydrates. It has a very thick basement membrane and has abundant collagen type 4 and glycoprotein. So that's the histology of the lens capsule. Then the subcapsular epithelium contains some cells which are mitotically active, so they're able to divide. Then they move towards the equator of the lens and transform into lens fibers. So the subcapsular epithelium has cells that are mitotically active, divide and move to the equator of the lens, then transform to lens fibers. Then finally the lens fibers, these are nucleated. Then they're deep to the cells of the subcapsular epithelium, and as you keep adding new fibers, they're added towards the periphery. So the deeper part of the lens, the fibers are loose, and the lens fibers are loose, and the nuclei keep on hardening. So the lens fibers have few cytoplasmic organelles, and they're usually filled with crystalline proteins. And these crystalline proteins are the ones responsible for transparency and refraction of the lens. So the lens fibers are produced throughout life, but as you keep aging, the production of lens fibers is less. Okay, so they have less organelles with crystalline proteins, and these proteins are the ones ensuring transparency and refraction of the lens. So you have a lens capsule, subcapsular epithelium there with mitotically dividing cells. Then they move to the equator and form the lens fibers. Again, the lens capsule, mitotically dividing cells in the subcapsular epithelium, then you have your lens fibers. And that's just cataract to show you where you have a cloudy lens. So next, we discuss the retina.